My name is Hengshuo Liu. I'm the Technical Sales Specialist from Agrobio System. It is my honor to join the DDW webinar. In today's presentation, I will discuss the challenge of cancer immunotherapy, how to overcome the obstacles, and the future opportunities. Cancer immunotherapy is a therapeutic approach for cancer treatment that uses patients' immune system to prevent, control, and eliminate cancer. It comes in several forms, including immune checkpoint inhibitor, T-cell transfer, monoclonal antibody, cancer vaccine, and immune system modulator. Cancer immunotherapy originated in the late 1890s when Dr. William Coley discovered that infecting cancer patients with certain bacteria sometimes resulted in tumor regression and even some complete remissions. In 1990, the FDA approved the first cancer immunotherapy, a bacteria-based tuberculosis vaccine called BCG, which was shown to be effective for patients with bladder cancers. During the past few decades, research has improved our knowledge of the mechanisms that regulate the immune system's response to cancer. However, challenges still exist, including the inability to predict treatment efficacy and patient response, the need for additional biomarkers, impaired efficacy due to tumor heterogeneity, the development of resistance to tumor treatment, autoimmune reaction, and high treatment cost. To overcome these challenges, researchers should focus on the following areas. Identify new targets that are specific to cancer cells and can be targeted by immunotherapy. Developing biomarkers that can predict and monitor patients' response to treatment. Improve combination therapy. Personalized treatment that can be tailored to each patient's unique tumor and immune system. Develop next-generation CAR T cells that can target a wider range of tumor antigens and can overcome immune-suppressive microenvironments. Promote cancer immunotherapy as an early administration to restore the anti-tumor function of patients' immune system. To better illustrate how to improve cancer immunotherapy, let's take CAR T as an example. So far, CAR T therapy has been only applied to hematologic cancer, which consists of 10% of the cancer diagnosis. There is an urgent need to expand CAR T therapy to solid tumor. Agrobio Systems offers over 50 types of proteins, including hematologic cancer markers such as CD19, BCMA, solid tumor markers such as HER2, EGFR, PSMA. Furthermore, Agrobio Systems has the platform to manufacture multi-pass transmembrane proteins, including CD20, GPRC5D, Claudin-6, Claudin-18, etc. The broader collection of CAR T targets will improve the cancer coverage of CAR T therapy. Expanding CAR T target is the first step for next generation of CAR T therapy. Clinical treatment with CAR T faces the challenge of antigen escape. In acute lymphoblastic leukemia, ALL, only 50% of pediatric and young adult patients treated with CAR T cells will be alive and disease-free 1 year after treatment. For patients with high disease burden at the time of treatment, the survival at 12 months dropped down to 31% due to CD19 downregulation. ALL patients who were treated with anti-CD22 CAR T cells after relapse with CD19-directed therapy achieved a 73% complete response rate, but two-thirds of these later relapsed with reduced CD22 expression. This challenge reflects the heterogeneity of cancer and how dynamic cancer cells are. Two strategies are proposed to overcome antigen escape, including co-administration of two CAR T cells specific for two different targets. The T cells can also be transduced with two CAR vectors encoding for two different targets, or transduced with one CAR construct with two single-chain fragment variables. These strategies can also overcome the challenge of tumor heterogeneity. So far, CAR T cells are engineered from patient-derived circulating T cells, so-called autologous CAR T. Although avoided of cell rejection, autologous CAR T limits collection efficiency, which is one of the reasons for its high cost. Next-generation CAR T therapy aims to expand the T cell source by establishing allogeneic CAR T. The T cell source will include circulating T cells from healthy donors. T cells differentiate from iPSC or stem cells isolated from umbilical cord blood. These allogenic T cells pose a risk of causing T cell graft rejection and graft versus host disease. A potential solution is the removal of TCR and beta microglobulin by improved gene editing technique. Among the allogenic T cell source, iPSCs have great potential in cell therapy for their multilineage differentiation ability. Besides of T cell differentiation, iPSCs are also used for natural killer cells and macrophage differentiation, which are important source for CAR-NK and CAR-macrophage that will further improve cell therapy options. iPSC culture requires critical microenvironment that can be formed by base membrane extract. Base membrane extract is collected from extracellular matrix-enriched mouse tumor cells. It is composed of laminin, type 4 collagen, nestin, glycoprotein, and different cytokines. By modifying the components, base membrane extracts provides different microenvironments for iPSC expansion and differentiation. For its fundamental function in iPSC culture, basement membrane extract will play an important role in next-generation CAR T therapy. Following the workflow of CAR T therapy, after identifying the target, clone the CAR, isolate the T cells for CAR engineering, the next important factor for CAR T therapy is cytokines, which is indispensable for CAR T cell expansion. During this step, engineered CAR Ts are expanded in the lab to millions of identical copies that are then infused back to patients for cancer therapy. Cytokine products are required at different stages during CAR T development. Acrobio system provides different grades of cytokine for specific needs. The RUO grade active max products are manufactured at ISO 9001/13485 certified facilities with research grade materials. Final products are sterilized by filtration. They are cost effective for exploratory studies during research and development phase. The premium grade products are manufactured by the same production method of GMP grade. They are animal origin free and have comparable quality to GMP, and have similar pricing to RUO. These products can transit to GMP grade seamlessly and are suitable for preclinical phase. The GMP grade cytokine products are required as raw materials for GMP grade CAR T expansion. Acrobio Systems provides strong support on cell therapy development with our product. We have broad coverage of target proteins, including more than 50 CAR T targets. We provide cytokines, activation beads, and basement membrane extract for ex vivo CAR T activation, expansion, and modification. Moreover, our antibodies and kits are powerful tools for detection of CAR expression, residue cytokine, and nuclease. In conclusion, cancer immunotherapy is a powerful and promising clinical approach, but facing many challenges, including unpredictable efficacy, developing resistance, high cost, etc. Future research should focus on improving cancer immunotherapy by enhancing safety and efficacy, improving combination therapy, and identifying new biomarkers. Thank you very much for listening. I'm happy to answer your questions.